I have your attention, please? All right, it's my great pleasure to introduce Dr. Paul Koch from UC Santa Cruz to deliver the first of two Ecology Center seminars this week. So we got my little cheat sheet with the abbreviated CV. So Paul has a bachelor's from the University of Rochester in geology and literature, I learned this morning. So we can talk books. He did all of his graduate studies at the University of Michigan, followed by a postdoc um, between the Smithsonian and the Carnegie Institute in DC. He's faculty at the University of California, Santa Cruz, like I said, where he's a distinguished professor. That just means old. That just means old. During that time, for 12 years, he was actually the Dean of Physical and Biological Sciences, and is currently the campus provost and executive vice chancellor of the whole university. Interim. There you go. Okay. Um, Paul has authored or co-authored over 150 papers, largely on the broad topic of chemical forensics, mostly isotope biogeochemistry, to study really applied to organismal remains, both modern and fossil, to study the habitats and diets and life processes of living and extinct animals. Um, Amongst all of his accomplishments, I was most impressed by the fact that Paul has graduated 20 PhDs in his time at UC Santa Cruz. And of those 20, 11 of them are faculty in academia today. And there might be a 12th soon, depending on the outcome of an interview. He's a fellow of the Paleontological Society, the Geological Society of America, and the California Academy of Sciences. So with that, let's welcome Paul. So I'll let you detangle.
Yeah, I will absolutely detangle. Can y'all hear me? Okay, yeah, great. Well, it's a real pleasure to be here. I was honored to be invited. I think that the grad students played a role in this. Is that correct? Yeah. So I told the chancellor I was giving a talk. I was going to be out of town because I was giving a talk that I'd been invited to by the guys who said those are the best talks to give. Like, so she was really happy and pleased that I was ditching the campus for several days, and then I'm going to ditch my phone for several days and go backpacking somewhere south of Salt Lake City. I don't know. So, um, so today I'm going to talk about, this is the first time I've ever given this talk. So it's a little bit of a test drive. It's a project on bodybuilding, not weightlifting, but literally building the bodies of animals and the role of the gut microbiome in that process, which is not something I thought I would ever work on. Oh, and I should say before I go further that doing this work, Angelique Martinez is a graduate student at the University of New Mexico, amazing PhD student, and Seth Newsom is her advisor, who set her on her path. And he was also a PhD student of mine back in the day. So when you think about an animal like that water buck over there, you know, a lot of times we think about how it makes a living. We think about the fuel. You know, it's eating this grass. Oh, sorry, I'm pressing the wrong thing. You know, it's eating grass. That's complex carbohydrates. It's hard for the water buck to digest that stuff, and it's going to get energy out of it. And I think we all know that for big hooved animals like this, their gut microbes are really critical to breaking down the fiber and releasing energy for them to fuel their bodies and to build some of the carbon parts of their bodies. But what we pay less attention to, at least I was paying less attention to, is the actual, it's mostly carbon. It's a carbon-based body, but it's got a lot of minerals in it. It's got bones so that it can stand up. Its body is made of protein. Okay, the bulk of its body is made of protein, and that's a carbon-nitrogen compound. It's not a carbon-oxygen compound. It's not a carbon-carbon compound. And the plants it's eating have a few percent nitrogen, and that body is 15% nitrogen. And so to turn this into this requires a trick of stoichiometry. And it's not, the oxpecker here has it much easier. It's pecking a bug off the side of the water buck. It's eating something that's rich in nitrogen, but the water buck is not. So we're going to talk a little bit about, what is the nitrogen that the animal is building its body with? Well, that's collagen, the most dominant protein in a vertebrate's body. And this is the amino acid composition of that collagen. So the collagen's a long polypeptide chain, actually 3 of them, woven into a triple helix. And it has actually a pretty peculiar amino acid sequence. And if you just looked at the, kind of the percents of each amino acid in the collagen molecule, it has this distribution. So the animal doesn't just need to just get any old protein. It needs ultimately it needs to meet its amino acid needs. It has needs for particular amino acids to build its body. And stop doing that. And so before I dive into the next complication, I just want to remind everybody about what an amino acid is. Central carbon, carboxyl group, amine group, and then this R group here that gives each amino acid gets its own name. Alanine here, threonine there. The R group is kind of a complicated dangling part that hangs off of the central carbon. And as it turns out, some of the R groups animals can make, and some of the R groups animals can't make. And so the essential ones are or they can't make these, or they can't make them fast enough. So they may have some of the biochemistry, but they are not able to make them fast enough to meet their needs. And these are groups the animals can make. So the problem for an animal is actually they have to eat these things, right? They have to get enough of these from their diet. That's why vegetarian humans worry about the amino acid balance of their food, because there's things they can make. And I'm showing you now the amino acid composition of the collagen protein in blue, and in green is an acacia leaf, because I'm going to show you elephants, and elephants eat acacias. So this elephant is lucky because actually the essential amino acids here are in greater abundance than in the collagen molecule. But it's a good thing it can make hydroxyproline and proline, and it's a good thing it can make glycine, and it may not make them fast enough. So the bulk of this talk is really about the role of the gut microbiome in helping the animals meet that balancing problem and getting the protein that they need. And what I'm going to end by saying is that for these large ungulates, for many of them, there is no such thing as an essential amino acid. Their microbes can make all of them, and they get them from their microbes. So that's the take home. So here's a host of microbes. So far in the work that I'm going to report to you, it looks like the dominant player are the bacteria that are in their guts. And many of people have thought generally about the beneficial effects of the gut microbiome. Maintains just general homeostasis, or stimulates the immune system, detoxifies plants, helps in recycling nitrogen. We'll come back to that. Synthesizes a bunch of stuff. And in particular, we've already talked about this, fermenting complex carbohydrates into fuel, which are simple fatty acids. But yeah, and we've talked about that. They digest the fiber and make energy. But the thing that I want to focus on is their role in synthesizing amino acids and recycling nitrogen. Didn't know that part was animated. So there's been a hint from laboratory experiments that largely using artificial tracers, so labeled substrates with extra nitrogen 15 in the substrate, there's been hints that some of the amino acids in an animal's body are coming not from the food they put in their mouth, but from the microbes in their guts. So some of the carbon skeletons And this is a study where this wasn't a diet study. They were just injecting mice with labeled substrates and seeing which essential amino acids were not coming from diet, but were coming from these labeled substrates through the microbes. So they were feeding the gut microbes and trying to see if the gut microbes label then showed up in the body protein of the animal. This is complicated work. It's done on rats and mice. There's some work on animals that are domesticated animals that are used for agriculture. There's a bit of other labeling work there that's shown some of this. There hasn't been that much evidence of large-scale uptake of gut microbe carbon into the animal bodies, but it's known to occur. And if you read any animal physiology text, it will say it happens. It's just not quantified. They know it. Like, why would you have a gut full of microbes and not eat some of the microbes, like when they die? Like, why would they be all lost through excreta and have no impact on the nitrogen budget? Why would it be only the exudate of the bacteria that would end up in the animal's body? Why wouldn't the bacteria end up in the animal's body? But there hadn't been a way to measure this in the field. And that way appeared, the path to this appeared the initial steps appeared 20 years ago, and the sort of the final work was in the last 5 years. So the key to this is that the essential amino acids, and here's 6 of them. The essential amino acids, when made by different producers, aerobic bacteria, anaerobic bacteria, fungi, plants, generally, I'm just going to call them green plants, the carbon isotope concentrations in the different individual amino acids are sorted in different ratios. The carbon 13 to carbon 12 ratio differs between, and these are normalized so they can be compared among all these samples, but you can see that the fungi have really high carbon 13 concentrations in isoleucine relative to the other producers. Whereas for leucine, anaerobic bacteria or aerobic bacteria have really high carbon isotope values relative to the other organisms. These are the producers. These are the things that actually can make these essential amino acids. So there's a very consistent pattern in different producers in how they sort carbon isotopes into these essential amino acids. And because the animals can't make these things, that pattern is preserved as you go up the food chain. So I've seen papers on sharks at the top, 5 trophic levels up from the top of a marine producing food web. And you can tell that all the carbon in that shark came from seagrass. Whereas all the essential amino acids in that shark came from seagrass. Whereas that other shark that looks just the same is swimming around, all of its amino acids came from advected phytoplankton that were blowing onto the reef. They were washing onto the reef. I'm going to do something much easier. I'm just going to look at herbivores, okay, and ask, like, 1 trophic level up, is there importation of fingerprint? Can we use carbon isotope fingerprints in these essential amino acids to track their importation up the food chain from the initial primary producers? And in our case, the producers are going to be bacteria and plants. It's going to be a simple system. And I should say that, animals eat a mix of things. No animal gets all of its amino acids from microbes or gets all of its amino acids from plants. So we're going to use multivariate statistics to deconvolute the mixed pattern of these isotope differences. And the reason I showed you the R groups is what I'm going to show you is this is about the R groups. Okay, the nitrogen, of course, is all coming from dietary protein in some way, and nucleic acids. But the R groups, the things that label those amino acids, which can be a lot of the carbon, the question is, where is that synthesized? Is that synthesized in a bacterium, or is that made by the animal? Or sorry, is that in the food the animal ate? Okay, is it clear? Any questions about this? Because this is okay. And this is just to show, this is, I believe, the first, oh yeah, go ahead. Plants, right. Yeah, it's a very good point, and I struggle with the word for that, right? Producers of essential amino acids, right? So fungus produce them by fermenting something, right? And bacteria, but we're talking about the producers of the amino acids. It's a really good distinction. So this is an example that Seth and one of his grad students, Alexi Besser, produced from the Sevieta Reserve in New Mexico, LTER site. They characterized the C3 plants. We'll talk about them more in a second. C4 plants here. And then they extracted microbes from the feces of desert rodents and characterized the amino acids in that feces that weren't mice cells. And they looked at kangaroo rats, deer mice, Peromyscus, and onychomemes are grasshopper mice. I think of them as wolves. They're like little carnivores. And what they saw when this is and I'll talk about the multivariate statistics they used in a minute, but they saw that the mice were falling in this mixing space between these three end members. And they used mixing models to calculate the proportion of the essential amino acids in the blood of these mice that came from gut microbes. And it was 40% for the deer mice, lower for the Peromyscus, and actually a little higher for the grasshopper mouse. And then this is the amount of the protein. This is the amount that was coming from diet from either C4 plants or from C3 plants. And look at the error bars there, because I'm not going to show you error bars on the rest of these figures. But we're talking about plus or minus 10% on this, 10 to 15%. This isn't like a spot measurement. So the goal here is to use this fingerprinting approach to look at large mammals, like that zebra, and to see how much of the protein, the essential amino acids in their body are coming from gut microbes, and how that varies with diet or how they digest their food. And then, because I'm a paleontologist, I don't work on problems unless I can do something on fossils. And so the end of this talk is going to be about Pleistocene vertebrates using the same approach, both extinct and extant vertebrates. So the first part is about animals from Amboseli Park in Kenya. I worked there a long time ago, 1990, 1993, studying bone decomposition. And in the end, I was studying actually elephant ecology that mattered for the ivory trade, or not having an ivory trade, more to the point. And Amboseli is a tropical savanna, extensive C4 grasslands, and there's shrublands and forests. And the forests are largely made up of C3 plants. We'll talk about these in a second. And the grasslands are largely C4 plants. And it's a beautiful place. That's Mount Kilimanjaro in the background. And so the plants in Amboseli, I collected plants when I was there long, long ago. A variety of trees and shrubs, acacias and palms are the dominant canopy formers. Diversity of shrubs, and then there's sparse forbs, like not much in the way of edible vegetation at ground level that's not grass, but there are some forbs there. And they're all C3. And C3 plants, when you look at the carbon-13 to carbon-12 ratios in them, they have relatively low concentrations of carbon-12. We're going to measure those differences using this delta value here. This is just a deviation relative to a mean. The lower the value, the more carbon-12. The way I should say it, the lower the value, the less carbon-13. The higher the value, the more carbon-13. Carbon-13 is the rare isotope. Nitrogen-15 works the same way. The higher this number, the more of the rare heavy isotope. The lower the number, less of that rare heavy isotope. What we've known for 50 years is that plants using the C3 pathway have less carbon-13 in them, and plants using the C4 pathway have more carbon-13 in them due to the way they do photosynthesis. And plants using the crassulacean acid metabolism pathway, CAM plants, cactuses, succulents, they use a pathway like the C4 plants, and they have higher carbon isotope values. And in Ambocelli, I point out there's this one. And the C4 pathway is largely in monocots, so grasses and sedges, and then in a variety, it sparsely occurs in some dicot plants. And there is this dicot bush here, Sueda, that I highlight because it's got C4 photosynthesis, but it is a food plant for Ambocelli animals. I'm not going to talk about it more because I still haven't sorted out what's going on with it, whether it's affecting the data that we're seeing, but it is the slight complication here that C3 and C4 doesn't just mean trees and shrubs versus grass because of Sueda. So that's the plants. I'm not giving you the list of their names, and then this is the cast of characters with respect to animals. We tried to shoot for 10 each of these for the study. We're a little short on the zebras at the moment. But there's a range of taxa from bovids here, various flavors of gazelle, the African buffalo, giraffes, hippopotamus, warthogs, elephants, rhinoceros, and zebras. I should say that the project I was doing in 1990 was a taphonomic project for all of the fossil lovers. I was studying bone breakdown. And, these rhinoceroses, one of them had died in 1960, okay? And it had been it was a famous rhinoceros in Ambocelli, and people knew where it was. And so these animals from the decay of their bones, we could tell that these have been on the landscape for anywhere from 30 to 15 years. There were freshies too for the wildebeest, but in general, some of these were all natural mortality. And then there's two ways that we're going to talk about these animals. One is that they're all herbivores, of course, and one is whether they eat grass, grazers. They browse. That means they eat leaves and twigs of the shrubby or tree plants. And mixed feeders, not omnivores. Now they're not eating meat, but they eat a mix of grass and browse. And we'll see these different dietary categories. Wildebeest are grazers, black rhinoceroses are browsers, elephants are classic mixed feeders. And then how they digest their food, whether they digest their fiber now in the foregut, in their stomach, or whether they digest their fiber in their hindgut, in the cecum, or in their colon. And hindgut fermenters, the primitive pathway. Food goes in, gets chewed, material gets broken down here with acids in the stomach. There is a lot of soluble compounds that are released by this process, get taken up in the small intestine. The fiber makes it down to this point, and then the microbes digest the fiber. There's more delivery of volatile nutrients into the animal's body off of the hindgut, off of the colon and cecum. The foregut fermenters, the ruminants are the cud chewers, the ones that chew their food twice or more. Food gets passed down to their stomach. It gets fermented here. It can't pass out of here. Well, I don't think it can pass out of here until it's at a particular particle size. There's a mesh here. And so that's why they keep rechewing it until they break down the particles. But there's a lot of fermentation products that are released here. The fiber is broken down. And there's lots and lots of absorbance of nutrients across the small intestine. There's a little, if any fiber makes it through, there's a little more fermentation down here. Urea is released off of the proteins that are broken down in this part. And that urea is recycled through the bloodstream back up into the mouth. Because when they rechew, they're fertilizing it. They're putting nitrogen back in the food. So it's a nitrogen recycling system that's super, super interesting and efficient. Nothing like that in the zebras, in the hindgut fermenters. So it's been understood that this is a more efficient way to ferment food and release energy. This approach is understood to be important for the release of energy from fiber. And we'll talk about the implications of that evolutionarily at the end of the talk. But I actually want to note the nitrogen cycle part of it here. Like that's a really efficient way to not lose nitrogen from your body, is to put urea in your mouth and make sure that you feed these bacteria, keep them well fed. So this is all the magic of Angelique. Well, not all of it. Okay, so I I demineralize the compounds and take a bone and get it down just to the collagen. Yeah, sure, I can dry a leaf in an oven. Okay, but then extracting the microbes from the feces using this technique described in this paper is a complicated thing. And Angelique has been doing that for little desert rodents and now for poos from a variety of large ungulates that I'll show you in a second. Actually, I want to go back to this slide and make a point. In these dietary categories that I'm showing you, browser, grazer, mixed feeder, these are what apply to large animals. If you're a small animal, like if you're this big, a small mammal this big, you can be more selective about the food you eat. And you can go after just eat the seeds, right? Like those rodents might just eat the high quality food, right? But if you're a big animal, like any of these animals, this is the only food that's out there in abundance to feed you. And so thinking about how you process this food is really the focus of this talk. There will be different stories if you get onto small mammals, herbivorous small mammals. Yeah. Oh, so we have we have an organic compound that's pretty purified. We break all the peptide bonds by hydrolyzing, so all the amino acids are just floating in solution. And then amino acids aren't volatile. They're not in the gas phase. So you slap a bunch of ethers on them, methane molecules on them at various points and make them volatile. They go into gas phase. And then you separate them using gas chromatography. And then as each molecule comes off the gas chromatograph, gets spit into a little furnace and combust into CO2, and then sent to a mass spectrometer, and cytosol value gets measured. And you measure everything in triplicate. So all the data I'm going to show you are three examples, three of these chromatograms that have been measured on a sample, the average value for three of them. And I wanted to just talk a moment about the fingerprinting, because I talked in general about what it is, but I want to dig a little deeper on it. The idea began with this paper in astrobiology, which is where all papers about mammal ecology start, by work done by Marilyn Fogel and James Scott and Diane O'Brien. They were the biogeochemists on this paper. Diane's at the University of Alaska. James and Marilyn have passed. But this paper laid out the idea. They were thinking about finding, bugs on Mars or something, right? And how we know what kind of bugs they were. So they were looking at all varieties of bacteria, and they noticed these patterns were appearing in the groups of bacteria. And it was really Diane who had the insight that these patterns might go up the food chain. And I think she published she began to look at other kinds of systems, and maybe that's why she got into the National Academy. So Marilyn got in for just volume on different kinds of things. But I think for Diane, it was this paper and really working on pivoting to work on nutritional ecology of Inuit people that are the reason she's been coronated or whatever they do to you when you go to the National Academy. So that was the beginning of this work in 2006. And I wanted to just basically what we're doing. So for my study, it's just going to be gut microbes versus plants, okay? So it's going to be, let's say the amino acid, let's just say it's delta 13C of isoleucine in gut microbe versus delta 13C of isoleucine in the animal, or let's C3 plant versus C4 plant, right? And what you can see is in bivariate space, there's clear separation here, right? But it's not aligned with the X and the Y axis. So all that this linear discriminant analysis does is it identifies that axis of first variation, and it just flips it and makes it the dominant and makes it the first linear discriminant. And any sample that was in that blue ellipse or in that yellow ellipse, they plop down here onto a single axis. And what I'm going to show you is that kind of approach, not in 2 dimensions, 3 dimensions, but in 5 dimensions, because there's 5 amino acids. And I'm going to show you the first discriminant because that's where most of the story is for these samples. So what you're going to see is a series of these histograms, one for gut microbe and one for plant. And then you're going to see where different animals fall relative to these histograms? Do they plot? Because remember, the animals don't make any of these things. They don't make any of these things. So if they plot on plant, it means they get those amino acids from plant. If they plot on microbe, it means they get those amino acids from microbes. I mentioned the sources. So the Ambicelli plants I just showed you are the source of the carbon isotope, the essential amino acid data for the land plants. The fecal microbe data come from large ungulates at this wildlife safari park in Oregon. So that's an entirely C3 ecosystem, doesn't have a mix of C3 and C4 plants. But it turns out that the pattern, there's very little difference in the sorting of isotopes into essential amino acids by C3 versus C4. The story is there's a little bit of signal there, but not much. So we're just going to combine the C3 and C4 plants. We normalize all the data. So every single one of these pathways I'm going to show you is normalized relative to the average amino acid composition of all the essentials. So it's always going to be a deviation relative to that average. So you can compare things that have really different carbon isotope values. And yeah, so here's Ambicelli, and that is the zoological park in Oregon, where you expect to see zebras running wildly. And these are the samples that we got from the various animals at the zoological park. So these are our sources. And this is an example of, this is the list of the organisms. Some occur in Ambicelli, some don't, but they're all large animals that ferment their food to get the goods out of them. And these are not normalized data. So you see they have different mean carbon isotope values. These are leaves from Ambicelli. The amino acid values of valine for leaves from Ambicelli, leucine, isoleucine, phenylalanine. And these are those gut microbes, not normalized. So we're seeing the raw data off the instrument. The normalized values would just be deviations from, small deviations from zero. And here are the smooth histogram density plots, if you like. If you fall in here, you're getting your amino acids from gut microbes. Fall in here, you're getting your amino acids from plants. I want to go to bulk data before I switch to the microbe data. So for most of my career, these are the kind of figures I would make. I would get the protein out of an animal. I'd measure its carbon isotope values of its bulk tissue. I'd measure the nitrogen isotope values of its bulk tissue. And I would interpret them the way I'm going to interpret right now, right? So the animals that eat grass have high carbon isotope values. They're C4 feeders. And here you're going to see that's what the grass feeders are always going to be in green. Wildebeest, Grant's gazelle. Well, some of the Grant's gazelles, you see the Grant's gazelles can do it all. Grazing, browsing, and mixed feeding. Thompsons can do grazing and mixed feeding. The impala is a committed mixed feeder. It's number 4 here. The buffalo is solidly a grazer. The giraffe is solidly a browser, eating C3 plants. The black rhino is solidly a browser, eating C3 plants. The elephants, where are they? 9 and 10. The elephants can either browse or they can be mixed feeders. They can eat more grass. Really, these are older elephants from around the time that the park was established, when there were a lot of trees in it. These are later elephants from the park after the elephants concentrated in the park and deforested it in a pretty serious way. And then I'm also showing you by the shape of the symbols whether they're hindgut or foregut fermenters. And so this is classic sort of savanna grassland. So between C3, C4, and mixed feeders, the nitrogen axis is a mix. It's a mix of where the animal was feeding. The wetter places tend to have plants with lower nitrogen isotope values, delta N15 values. And subtly, I'm not going to go back to the plant data, the nitrogen fixing plants, like the acacia, have lower nitrogen isotope values than the plants that don't fix nitrogen. So feeding on grass, which doesn't fix nitrogen, may give you a higher nitrogen isotope value than feeding on acacias, which do fix nitrogen. So that's kind of the standard approach to these data. And yeah, the main point to note is that we're going to classify these animals not just by their name, but we're going to classify them by their diet. And if they really do mix it up, We're going to split their data up to recognize those 3 different diets. And we're also going to classify them by how they ferment their food. These are all from, no, these are bone data. This is from the bone of, these are from the bones of the animals.
Their bones are growing. And then a different gazelle only graze?
Yeah. So these are all the data right now. Like I said, we're trying to get 10 individuals. So because we have enough individuals here, we can break the gazelles up into the ones like this individual, these individuals here. Well, actually, for the Grant's gazelle, there's only one Grant's gazelle that falls solidly in the grazing category. It's that individual right there. Most of the Grant's gazelles, you see these number 2s in here, they're mixed feeders, and they were mixed feeders for the interval of bone formation, which is like 7 years to form the collagen in an adult animal's bone, roughly. So over that time period, their average diet was a mix of browse and grass. And we can't say whether that animal switched seasonally between all grass one part of the year and all browse the other part of the year, because it's bone, it's all mixed up, right? So it's the average, but it's a really good point. I showed a bone that I didn't say out loud. The animal data are coming from bones, okay? So bones have a residence, like collagen has a residence time of roughly 7 years in an animal bone. And so all of the data I'm going to show on the individual amino acid data is going to be the same story. These are the average diet of the animal for that window, temporal window. Any other questions at this point? Okay. So there are hundreds and hundreds and hundreds of papers like this that looking at carbon isotope values versus nitrogen trying to interpret animal ecology. So there are not hundreds of these kinds of papers, these kinds of figures. So here we're looking at now we've averaged together all the animals in each of these categories. So there are 15 different categories here. Wildebeest just graze, and so we're just showing all the combined data for all the wildebeest plot here in this LD score. Remember, this is for the 4 or 5. I can't remember how many she analyzed. I'm going to say 5 essential amino acids. This is where they plot in multivariate space relative to microbe versus plant. In the case of the zebras, there were only 3 individuals here. They've been averaged to create a mean, and then we've looked at where that mean plots in this linear discriminant space. And you can see there is some patterning here. There's maybe more squares over here, like maybe more hindgut fermenters here over the plant part, and maybe more foregut fermenters here over closer to the gut microbe part. But it's still a little mixed up. There are some patterns here that make sense, though. The browsing Grant's gazelle has more plant in its diet than the mixed feeding Grant gazelle versus the grazing Grant's gazelle. And I say that because this is the browse. I'm going to get to this later. Browse is higher quality food than grass. And so you probably have to ferment browse less than you have to ferment grass to get food out of it. So there are patterns that are emerging here that make some sense based on the quality of the food, or that the hindgut fermenters, which probably ferment their food less, have more plant in their bones than the foregut fermenters, which have to ferment their food hard to get resources out of it. So more gut microbes for these foregut fermenters. Going into this, our hypothesis was that the foregut fermenters would show this effect more than the hindgut fermenters. And that's beginning to emerge here. I warned you I wasn't going to show error bars, and so just roll with me on that. This is the mean for the microbe histogram, and these are the 95% confidence interval. So giving you some rough sense of, these values here really, and this is the 95% confidence interval for its microbes came from plants. So these values out here are really getting out to the statistical edge. They really do look like these must be largely gut microbes. Whereas these values here are sitting at the mean for plants. So maybe this hippopotamus is a plant eater even though it's, even though it's way out on the, yeah, even though it does ferment its food in its foregut. So you can take these kinds of data where you've got two sources, gut microbe and plant, and you've got error on them. And each of these dots has an error on it that I'm not showing you, because this is a mean for, in this case, I think we had like 11 rhinos. So this is the mean for 11 different rhinos. There's error associated with that, and there's error associated with the measurement. And so you can use Bayesian mixing models, isotope mixing models, that take account of the error in both the sources and the consumers. And you can calculate percent microbe versus percent plant by animal, or by the average for these animals. And again, those percentages, that's really what, in that Besser paper, I showed you the little bars with the error bars on them. That's what Besser had done. She was just plotting them in a different way. And so what you see is that for when you do one of these mixing models and look at the proportional contribution, there are two patterns that jump out to me. So here are the browsers, here are the mixed feeders, here are the grazers. And what you see is that as you move across that diagram, there's more and more of that pale tan color. That's more and more contribution from gut microbes. So we'll talk about why. So that's a diet signal. Like why is grass leading to greater microbe contribution? The other pattern here is within each of these categories, hindgut browsers versus foregut browsers, hindgut mixed feeders versus foregut mixed feeders, hindgut grazers versus foregut grazers. And the general pattern there is that if you're a foregut feeder within a diet category, you have way more microbes showing up in your bones. Way more microbe-generated amino acids are showing up in your bones. Hippos are an anomaly, and that is true in almost every isotope study that I've ever done. For any system, hippos are always wrong relative to whatever your theory is. And I don't have an answer for why hippos are so wrong so often, but inquiring minds want to know. When I showed these data to a friend, he said, "Well, of course they're carnivorous. They're just eating people." I don't know. Or maybe they're eating fish. I don't know. It is true for calcium isotopes. Hippos look like they're carnivores. They plot with the lions. So just going to say that. So what's the diet driver? The diet driver, there's two possibilities. So why are the grass feeders that happen to be C4 plants, why are they doing so much synthesis, microbial synthesis? And there's two ideas here. One is that there's just less nitrogen in the C4 plants. So this may somehow relate to nitrogen recycling or the quality of the food. The other idea is that C4 plants have higher fiber content. Because of the way they do photosynthesis in these bundle sheaths, they actually are harder to break down. And so it may be that to get food out of the C4 plants, you just have to ferment them longer and harder. So there's an explanation based on these two contrasting, I would say, diet-driven explanations for what's going on, for why we see a diet signal in the ambicelli animals. The hindgut foregut hindgut fermenters are just less efficient at getting fermentation products out of their food because of where they ferment it. So we thought we were going to be first to this, but Diane, whose picture I showed you, published a paper last week doing this kind of work on moose and muskox. And she's seeing roughly 50% balance between microbe-generated amino acids in the bones of moose or muskox, and 50% of the amino acids are coming from diet. So about half and half. The rabbits here are a little more, but they eat their poo a second time. So they're coprophages. There's like poo number 1 and poo number 2, and you can tell it apart. So, and so it may be that coprophagy is leading to the greater microbe contribution in the snowshoe hares. So anyway, so the take home here is that there's substantial incorporation of microbially synthesized essential amino acids in many large African herbivores that live on savannas. That digestive physiology matters. If you ferment your food in your hindgut, You have less microbes than if you ferment your food in your foregut. Diet matters. If you eat C4 plants, you eat C4 grass, you do more fermentation, you get more microbes in your bones, in your bone proteins, than if you eat C3 plants. The drivers of that dietary variation could be nitrogen concentration, could be fiber, could be something else we haven't thought of. The gut microbiome research has largely focused on fiber digestion and energy generation. And the main way in the papers that I've looked at, okay, so I'm a humble paleontologist, like I just started reading this gut literature on nitrogen. Most of the papers are about the role of nitrogen limitation and limiting the microbiome. They're not about the microbiome as a source of amino acids for bodybuilding. There's beginning to be a change in this literature. Like, hey, it actually looks like there's a lot. The gut microbe isn't like food limited, which isn't surprising given that the microbes are inside of a giant bag of protein, that's shedding cellular material. Like the gut sheds a tremendous amount of material. It was hard for me to imagine that it was nitrogen limited, but that is the predominant paradigm. But our work suggests that the microbes play a critical role in protein metabolism. And likely recycling, and that, animals are building those bodies, the protein in those bodies, a lot of those amino acids are coming from gut microbes. The R groups are coming from gut microbes. They're not coming from the food they put in their mouths. And remember, these are only the non-essentials. These are only the essential amino acids that we're looking at. We still haven't figured out how to look at the non-essentials in the field. But when you look at non-essentials in the lab using tracers, you can see that they do the same thing too. They turn carbohydrate into amino acid. So they may be doing it in their own bodies. It may be that their gut microbes are doing it for them. But either way, they're turning sugar into protein. So this is a big deal. So I've left myself with 10 minutes to talk about the Pleistocene. That's okay. That's okay, because it's pretty much the same story with the same approach. Here are where the animals come from. Michigan, where I did my grad school. Iowa, where I had a colleague who sent me a group of samples. And Alaska, where my student Kana did some of her PhD work, and we had little bits of animal lying around the lab. And these are from the last glacial maximum to the late glacial period. These are not savannas, okay? These are steppe tundra, boreal forests. All C3 plants, no C4 plants. Here are the players, broken down the same way: hindgut, foregut, grazer, mixed feeder, and browser. But remember, grazer here doesn't mean C4 grass. Grazer means C3 grass. Here is a bivariate plot of the sort that I just showed you. It's extremely messy, okay? Like searching for pattern in this thing, like Pleistocene ecologists spend a lot of time trying to explain this kind of stuff, right? Like, maybe there's more, trees in the diet of this thing, and there's more grasses but honestly, like these mammoth data here and that one horse, the cave lions from this site have nitrogen isotope values like that. And the bears from this. So these are very weird nitrogen isotope values. In any case, especially on the carbon axis, we're thinking about like the source of carbon into the system. There's quite a mix here across the C3, across this admittedly very small gradient here. We're only looking at a 4 per mole spread. In the C3, C4 plots, I was showing you 12 and 15 per mole spreads in carbon. So this is just the C3 end member. So teasing apart diet here is pretty tricky. But people do it. So no strong partitioning of food by taxon. And yeah, I don't know what's up with the mammoths. I don't think they're carnivores, but if you just classify them by diet type, it's a little bit better. So now everything that eats grass is in green, everything that eats browse is in brown, and the yellow are the mixed feeders. It's a little more clustered, but more in the nitrogen space than the carbon space. More on the nitrogen axis than the carbon axis. So not much information here that seems to be coming in about diet, and especially this question of like, where do they get their protein from? No information. So we're back to these kinds of diagrams. These are all the specimens, because we don't have as many specimens as for the African example. And it's when you're just looking at, hindgut, foregut, the kind of species level data, it's pretty messy. So the mammoths are grazers. Yeah, they're more towards this end. If grass is hard to ferment, maybe that explains why they're more at this end. And the bison are certainly grass eaters. The mastodons they eat browse. They're the kind of elephant that eats just leaves and twigs. So there's some pattern here that makes a little bit of sense, but let's just say at the individual specimen level, not as much sense. We go to the species level. And this is the same data set, those same error bars apply to these diagrams. So the mean is here, the mean is here. The 95th percent confidence interval for the gut microbes is there. So you can see now that there is a cluster of animals here that could be getting gut microbe, pretty big gut microbe contributions. And there's this other group that are like way out in the plant. These are clearly plant microbe. Plants are ending up in their bones. But there's still not a diet pattern here, right? Other than these mixed feeders all are a little bit towards this end. But for the other diet categories, they're spread across a pretty wide range. For grazers and browsers, you've got some that look like they'd be getting gut microbes and others that look like they're getting plants in their plant amino acids in their bones. So, but if you just highlight what was there in the circles versus the squares, if you just highlight what was present in whether they're foregut or hindgut, you see the pattern here is if you're a hindgut fermenter, you're getting the amino acids in your bones are coming from plants. And if you're a foregut fermenter, they're mostly coming from microbes. Well, there's way more microbe contribution if you're a foregut fermenter. And diet doesn't matter. It's now what matters. Largely what matters is digestive physiology. And this is just showing, that's just cleaning that pattern up so you're not seeing it at the individual specimen level, you're seeing it at the species level, right? So it's very clear there that the hindgut fermenting species are planty and the foregut fermenting species have more protein in their bones is coming from the microbes. This figure, just like the last time, hindgut, foregut, only foregut fermenters for the mixed feeders, and then hindgut, foregut for the grazers. And yet, the diet signal is kind of, even without hippos, the diet signal is messed up here. The foregut fermenting sloth, when it's browsing, has like half of its amino acids are coming from its microbes. By the way, the first data on this for any extinct sloth, the fact that we know something about the digestive physiology of a giant sloth is kind of cool to me. The cool part of the talk emerges. The extinct helmeted muskox is not like the surviving tundra muskox. The mammoth, despite eating a lot of grass, gets almost none of its amino acids from its gut microbes, whereas the bison, lying next to it, gets a lot of its amino acids from microbes. But this just reiterates what that prior slide showed. What matters here is not diet. What matters here is whether you're hindgut or foregut. If you're a foregut fermenter, you have microbial contributions. Okay, so penultimate slide. Over the history of the age of mammals, since the dinosaurs checked out and the mammals took over, among the terrestrial vertebrates that don't fly, there's been a switch in digestive physiology between hindgut fermenting when in the Oligocene, and as the planet cooled down and as the environments changed, and especially as we came into the glacial age, the Pleistocene, and the recent, hindgut fermenters have essentially disappeared as species across the planet. And they've been replaced by foregut fermenters. And this paper by Christine Janis, which pointed this pattern out, really talked more about climate driving the energy part of that equation, like food availability and fuel availability. It wasn't about maybe the edge that these animals have, that the foregut fermenters have, that relate to nitrogen recycling. And being able to do away with the idea of having essential amino acids. that there's an edge that comes from building your body when the microbes are doing the amino acid balancing for you. And you don't have to do it by what you put in your mouth. So I think there's a lot of that maybe macroevolutionary interest that might flow out of, like I said, this is the first talk on this, this whole story, and we're still gathering. We have another, I'll end with this, what's up next? Well, for the Pleistocene work, we didn't use boreal plant and producers as our mixing space. We used the African plants. And so Diane, in that paper that they published last week, has 50 boreal plant taxa in it. So we're going to immediately assimilate those data and rerun this analysis using her data. And I didn't show you any samples from Pleistocene mammals that fed on C4 plants. There are Pleistocene mammals that feed on C4 plants. We know that from their stable isotopes. They all live in Missouri and New Mexico and Texas, not Michigan and Alaska. And so we have about 50 such samples in Seth's lab at this point that we're going to start extracting protein from to see if the real reason that diet doesn't matter is because the Alaskan ecosystems, these Pleistocene ecosystems, didn't have C4 plants. It could be that the key here is that if you really want to see a lot of microbial contributions to bone protein, you need to have foregut fermenters eating C4 grass. And just Diane's data didn't see a big microbial contribution either. It capped out at 50%, just like ours did. So it could be that that's because there's no C4 plants in these ecosystems. And we get into Texas and New Mexico and Missouri and start looking at bison or mammoths or camels. There are camels, of course, in North America. We start looking at those animals that we're going to start to see really high percentages of gut microbe contribution in the Pleistocene animals. So summary overall, this just said it. Yeah, we don't see body protein coming from microbes, more than 50% body protein coming from microbes in these animals. We do see that in Ambicelli, but no C4 grasses, no C4 grazers. Unlike Ambicelli, the strongest consistent determinant of microbial contributions to animal protein is digestive physiology, not diet, or not digestive physiology plus diet. The microbial generation of amino acids, especially in foregut fermenters, may have had a profound impact on their rise to dominance over the last 30 million years, especially with the spread of C4 grasslands and sloths, which are, they aren't as weird as hippos. Okay, the sloth data made sense. The hippo data, not so much. So I will stop there and entertain. I've used up all the time, but I'll entertain questions. I don't know if we're going to get kicked out of here or not. Yeah, go ahead. Yeah, thanks. Yeah. Right. Yep. Yeah.
Smaller browsers, you can be a lot more choosy about what plant parts they're eating. So
Yep.
something like an elephant is eating a lot more wood.
Knock the tree over, yeah. Yeah. So I think in this body size category, like these are all quite large. These are all mega herbivores in some sense, right? Almost all of them. The Tommies, maybe not, but everything else is really quite big. I think in this category, we wouldn't be seeing those body size effects. But as I mentioned at the start, as you start dropping, especially to, there's going to be gum exudate feeders. I mean, among the primates, there's all manner of ways of being an herbivore. Even in those rodents that Seth and Alexi studied, there are ways of making a living by being very selective that I think are gonna change the story a lot when you get to small mammals. Yeah.
Saying that, so in the contemporary samples, you have much less spread of the data and have much fewer samples that are solidly over, way into the plants.
In the Pleistocene samples, I have
Yeah.
solid, yeah.
Okay, so you have a lot more spread
Yeah,
there.
yeah.
Samples where they're really got a lot more.
Yeah, ask the total question, I'll answer. Yeah.
Part of what is driving that is that now there's so much more foregut digesters than hind.
I am saying that foregut fermenters are more common now, and that especially when they're on C4 grasslands, they get a lot of their body protein from the R groups, the amino acids in their body protein. A lot more of that is coming from microbial products than from the food they put in their mouths. And maybe the same thing will be true when we get to the Pleistocene, right? When we get to a C4 grassland in the Pleistocene, maybe we'll see the same pattern. I'm really anxious to get that. But in the long term, Oligocene relative to today, what I was saying is that this recycling and this ability to have your gut microbe do your amino acid balancing for you so that you don't have to be selective about what you feed to make sure you get enough phenylalanine in your diet. Like I did when I was a vegetarian, I was like worried about the amino acid balance in my food, right? That bison doesn't have to worry about it. Its microbes take care of it. And those animals are, macroevolutionarily, those animals are the winners, right? There's 5 species of rhino. There's 2 species of elephant. I'm not counting the genetic species. Okay, I'm just, sorry. There's I don't know how many tapirs, not many species of tapir. There's 3 species of horse, and there's how many gazelles? And how many the winners in this moment with respect to these large hooved animals are the foregut fermenters. They're the artiodactyls, right? Not the perissodactyls. And not the elephants, not the proboscideans. Yeah. And I think this may have something to do with it. Anything else? Okay, you guys can decide when I have to stop. Yeah, go ahead. Yeah.
Oh, it's weird. It's aquatic.
Yes.
Aquatic one.
Yeah. We haven't looked at tapirs yet, which are semi-aquatic. Elephants also can be pretty aquatic, but nothing like a hippo. I mean, it's really surprising. Hippos, the story on hippos is that all day they roll around in the water and they don't eat land, they don't eat aquatic plants. And then they come out at night and they go ranging across the grasslands, the savannas eating grass. And carbonostope-wise, that makes perfect, just bulk carbonostope data, that makes perfect sense. But every other proxy that we've thrown at them, zinc isotopes, calcium isotopes, now this, amino acid specific stuff, they don't behave like other herbivores. They're not processing elements and amino acids like other herbivores. And I don't know what it is. It may be aquatic vegetation, that they do eat more aquatic vegetation than we think, and that's affecting it. It may be that there's macrobionts on some of that aquatic vegetation that's affecting their amino acid balance, at least. It wouldn't explain the zinc, but we have seen something like this with sea cows, that modern fossil sea cows, they eat like a lot of kelp, right, and a lot of seagrass, but it turns out there's tunicates on that stuff, and occasionally their amino acid balance can be really affected by the occasional tunicate, the occasional animal that's on the plant that they're eating. So maybe that's what's happening in hippos, but I don't have an explanation for it.
One more. There's one more question.
Yeah.
Species or region.
Yeah.
And whether that would matter. Acid.
Yep. So Seth, in the work that he did with Alexi and on all the work that he does, he sequences the 16S RNA out of the feces. So he knows the bacterial makeup of who's in the feces, right? But he can't determine. He's only looked at 2 groups so far. He's only looked at the mice and now these animals that are out of their natural habitat, right, in Oregon, right? It is, and Diane, she used data on different kinds of bacteria from the literature, and then she had fistulated moose, so she took ruminant liquid out of moose, 5 different moose, and she isolated those bacteria, and she knew those were anaerobic bacteria. So the story of her paper, which is why it had that scintillating title, anaerobic bacteria explained, is because she figured out that it's the anaerobic bacteria, particularly, that are producing the pattern that, and she ran Alexi's data over using her training set, and she got the same answer as Alexi got using the poo microbes. So it is, the bacteria do matter, but which anaerobic bacterium does the species matter? What is a bacterial species anyway? But I do believe that that the composition of the microbiome may matter, and we have access to samples of bison that switch between C3 and C4 over the course of a year. They're seasonal. The early green up is C3, and then the late green up is C4 in parts of the Midwest. And those bison keep growing their tails through the whole time, or you can take a blood sample from them. So you might be able to see if this pattern changes, and if you measured the poos of the bison and looked at the bacterial composition, you could see if the microbe is responding to the change in the diet. But it's a great question, but with respect to this particular question, super early. Yeah. Okay. Thanks.
And here at my house tonight, our all welcome.